Welcome everyone. Thank you for joining us for the fifth lecture of Sciencia's 2021-22 lecture series. Welcome to those of you in person. Welcome to those of you on Zoom. We're delighted you're with us. I'm Anthony Brand, Professor of Composition and Theory at the Shepherd School of Music and Director of Scientia. Each year, Scientia brings together outstanding Rice faculty to examine broad themes that transcend disciplines. This year's series celebrates and explores the vast interconnectedness that animates nature and human life. This afternoon, we're excited to welcome engineer Lauren Stadler and biologist Scott Egan. Lauren Stadler is an assistant professor of civil and environmental engineering at Rice University. She's an environmental engineer whose research focuses on wastewater-based epidemiology, environmental antibiotic resistance, wastewater and resource recovery, and environmental synthetic biology. She has been collaborating with the Houston Health Department since the start of the COVID-19 pandemic to develop methods, implement, and operationalize a citywide wastewater monitoring system that informs public health action in real time. Scott Egan is an associate professor in the Department of Biosciences at Rice. He is an integrative evolutionary biologist whose research program spans many disciplines, including evolution, ecology, behavior, conservation, genetics, and genomics. His lab has been funded by the EPA, USDA, NOAA, and DoD to investigate the use of eDNA for the detection of invasive or rare species, characterize biological communities, and to study how eDNA can work as a detection tool in flowing waters, such as streams and rivers where the source of the DNA is far from the site of detection. We'll have 10 minutes for question and answers after the talks, and then invite you to our in-person reception here out in the lobby. Also, please mark April 26th, please mark April 26th at 7 p.m. on your calendars for our annual Bachner Lecture, which this year features Merlin Sheldrake, author of the wonderful book Entangled Life about fungi and mycelial networks. The free lecture will be held at the Glasscock School's Hudspeth Auditorium, and there'll be a book signing that will follow Dr. Sheldrake's talk. Please join me in welcoming Lauren and Scott.
Thanks so much for the introduction and the invitation to speak here and share some of the work that we have been doing in collaboration with the Houston Health Department across this pandemic that we've been living through. And I'll talk about kind of the history of our wastewater surveillance system here in Houston and what we see for it in the future for monitoring diseases. Okay, before I get started, I really have to emphasize how collaborative this project has been. I really think the success of this project has in large part due to the fact that we've had this collaborative and fluid relationship with our health department and with our utility here in Houston. The project is led by Dr. Hopkins at the Houston Health Department in collaboration with Dr. Enzer and myself here at Rice. And then Dr. Tregan has played a critical role in supporting the analytics on this project. And there's been a number of different folks who are involved, and you can see by the coloring of the names, the Houston Health Department folks are in orange, the Rice University folks in dark blue, and the Houston Public Works individuals in light blue. And all of these individuals are critical to generating information on a weekly basis that is actionable and has high statistical confidence. So I hope, and maybe I assume, that many of you have heard of wastewater surveillance before. It's been in the news. Many of you maybe had not heard of it before this pandemic, but over the course of this pandemic, it's really emerged as a tool for public health use. But just as an overview, it begins with a positive person, an infected individual, using the restroom. And it turns out that both symptomatic and asymptomatic individuals shed viral particles in their stool. And this has become a really reliable indicator of infection. And that stool enters our wastewater and enters the sewer system, where it then travels through our sewer system to wastewater treatment plants. And we can collect a sample of that untreated wastewater, and we can assay it to determine the amount of viral particles in that sample. And we're basically looking for tiny fragments of the genome of this virus. And so we've developed sensitive methods for detecting and quantifying the amount of virus in wastewater samples, and also methods for screening or understanding which variants are circulating in our communities. I'll talk about both of those different pipelines. And there's a number of reasons why people are super excited about this as a tool for public health. And those, some of the reasons are, first, as I already mentioned, it allows for the detection of both symptomatic and asymptomatic cases, because both, whether you're symptomatic or not, you can shed virus in your stool. It's very resource efficient, because wastewater represents this pooled sample of all the individuals that are contributing to that waste stream. It does not depend on individual testing and reporting. And especially if we think about over the course of this pandemic, how things have shifted. At the beginning of the pandemic, we would go to a testing facility, right, and get a PCR test. But now, if you are feeling sick and you want to test yourself, you're most likely going to go and get a home test, right? And that's not necessarily going to be reported to anybody. So that information is really not being aggregated in any way that, for example, our health department leadership can make decisions based on. It does not depend on health-seeking habits. Again, the people who are most likely to get tested are individuals who are symptomatic. And if you even look back at the CDC guidelines back when we were all going to facilities to get tested, not here on Rice's campus because we were all being tested regularly, but the general public was encouraged you were encouraged to go get tested if you were symptomatic. So all of the clinical data is very biased towards symptomatic individuals. And now there's a large body of research that shows that wastewater trends tend to emerge several days before other surveillance methods. And when I say other surveillance methods, I'm speaking about clinical surveillance, so PCR testing, hospitalizations, and even syndromic reporting. And so wastewater can really serve as an early warning indicator of infections in your community. Of course, we have to keep in mind that wastewater surveillance requires that you have a municipal wastewater treatment plant, that you have a sewer system that we can sample from and collect these wastewater samples. Not all individuals, especially as you go into more rural areas, are on a sewer system, right? And so this type of method wouldn't necessarily work in the same way in more rural communities that are septic. And then, of course, we have to keep in mind that a negative wastewater result does not clear a community of SARS-CoV-2 infections. There is, it's not perfectly sensitive, and that we would pick up every possible infection in the community by assaying the wastewater. And there's a lot of factors that will impact the sensitivity of this approach that I'd be happy to talk more about. So now I'll shift into talking about the wastewater surveillance system here in Houston. This is a map that shows all of the places that we're currently sampling. So we first started sampling the wastewater treatment plants in Houston in May of 2020. Since then, it has expanded to include a number of other sites, including schools, congregate living facilities, and lift stations, which are sub-sewer shed locations, so that we can break down some of these larger wastewater treatment plant sewer sheds into more resolved areas. Houston's kind of unique in that we have 39 wastewater treatment plants. Most large metropolitan areas are served by just a handful of very large centralized wastewater treatment facilities. So as an example, Chicago, third largest city, probably closest in population size to Houston, they have just 3 wastewater treatment facilities. Houston, we have 39, that's a lot. They range in size from serving less than 1,000 people to serving over 500,000 people. In many ways, this is a huge advantage when we think about wastewater monitoring, because we have these 39 locations, so we can get a geospatial look at Houston every single week just by sampling at these 39 existing locations. Okay, so here's an overview of what happens every single week. We get composite samples, which means we don't just grab a sample of wastewater directly from the sewer line. There are machines that are collecting samples over 24 hours to get a more representative picture of the wastewater from the community. Those composite samples are transported to my lab at Rice University and the Houston Health Department lab. We assay those wastewater samples to determine the concentration of virus in them. And we also enrich via amplification the genomes of SARS-CoV-2 out of these wastewater samples, and we sequence them. And that is used for screening for variants of concern. This information is input into statistical models that then identify areas across the city where we see increasing trends or emergence of variants of concern. And that information is summarized in a report that's sent to health department leadership every single week. And they use that information along with a number of other metrics to prioritize interventions at the zip code level. So they'll use the wastewater data to identify priority zip codes and where to send resources. And I'll give a couple examples of that at the end. This is just an overview of the analysis pipeline that we do. Again, there's kind of two different arms of this. One is the quantification of the virus, where we quantify the amount of virus in each of these wastewater samples. And then the second is how we sequence it to determine not just how much is there, but what is there, which variants are circulating. First, I'll talk about the quantification piece and show some of the data that we've generated. A really quick overview of the process, in part also because it's relevant to Dr. Egan's talk after, and that we use somewhat similar methods, basically detecting these fragments of DNA or RNA in environmental samples to tell us something about the community or the system that we care about. These viral genomes are present at very low levels or very dilute levels in our wastewater samples. So we have to do a concentration step to basically increase that concentration in our sample so that we can measure it. We did an extensive study comparing a whole bunch of different concentration methods to choose the method that we currently use based on what was giving us the most sensitive detection, highest throughput, lowest cost, most reproducible. We also did this for other steps of this process, for the RNA extraction and purification, and then also for the quantification to end up with the method that we're currently using. So what we do right now is we take 50 mils of wastewater, we add some salt to it, and we filter it through an electronegative filter that captures the virus on this filter. We then beat that filter or lyse, basically mechanically beat it up to lyse those viral particles and release the genetic material. Then we purify it, and then we quantify very small fragments of the genome. So now I'm going to start showing some of the data. Here is really a summary graph of all the data in one here. So here I'm showing the viral load in the green line, and it is relative to a benchmark level, and I'll talk a little bit more why we report it that way. So the benchmark level is July 6th of 2020, and then I'm also showing a clinical positivity rate in the blue line, which is the 14-day moving average of that clinical positivity rate, where each of the X's are the daily measurements. And what you can see here is these are strongly correlated with one another, they're moving with each other, and actually the wastewater data can actually be used to predict positivity rates because it's such a strong relationship between these two. And while this clinical data is showing it on the day that the specimen was collected, that's what each of those hatches are, in reality it actually takes a little bit more time for that data to become available to public health officials and cleaned and amassed. So while these dates are the dates these specimens were collected, that information is actually delayed coming in. And that just has to do with the fact that this information has to be aggregated across the entire city. It has to be cleaned, in that there are sometimes duplicates or people have been tested multiple times, right? And that data has to be cleaned such that the wastewater data actually comes in a lot earlier. We get the samples on Tuesday and we'll have the results by Thursday. And so this wastewater data can give an immediate picture of what's going on across the city. And we've done this not just at the aggregate, across all of Houston, but at each of those 39 wastewater treatment facilities. And we see this strong relationship across many of the facilities. So I'm showing here 6 different wastewater treatment plants that vary in size and flow rate of wastewater treated. I think it's, 69th Street is our largest wastewater treatment facility, which is why you see it's kind of the tightest data set. If you go to some of these smaller wastewater treatment facilities like Tidwell Timbers or WCID 76 that serve very few people, you see one thing to note is the positivity rate data is highly variable, just because not that many people are getting tested any given week. And so it's highly sporadic, right? And in those cases, the wastewater data is also more variable because it's a smaller community, but it's a much more reliable source of information because it's capturing, this constant stream of wastewater from this community week over week. And in many cases, it becomes the primary source of information for these smaller communities for understanding what's going on in terms of infection dynamics for the health department, because it is more consistent than their positivity rate information. Now I'm showing here a heat map of all the wastewater treatment sites on the y-axis and over time on the x-axis. And here I'm showing the viral load, which is the viral concentration times the wastewater flow rate, relative to this benchmark level. And so we decided to report it out as a benchmark level instead of a viral load because we realized that was better understood by health department leaders. When we talked about copies of virus per liter of wastewater, it was not something anybody had any tangible, nobody really knows what to think about. We think copies per liter, copies of a virus in a liter of wastewater. It wasn't meaningful to anyone. But the level of virus in the wastewater in our first surge, so July of 2020 was our first major surge of COVID across Houston. That was really understood by leadership, and so now we benchmark everything to that first surge level. And this heat map, very clearly shows the surges that have occurred since then, right? We're seeing the Alpha, the Delta surge, and the Omicron surge most recently. And you can see, I guess this is from, end of March. We're starting to see it's overall low across the city, but we are seeing a little bit of an uptick now. Now I'll transition to talking about how we screen for variants of concern, and this has been collaborative with Dr. Egan in the Department of Computer Science, and we have developed a pipeline to screen for variants of concern that have emerged by sequencing the viral genomes from these wastewater samples. And we've been doing this now for, I think, almost 50 weeks. So we have a lot of longitudinal data now. But one of the challenges of wastewater is that it's a mixture, right? It's a mixture of everyone that contributes to that waste stream, which means it's a mixture of all the variants that are circulating in the community. And if we want to use wastewater for an early detection tool to detect emerging variants that are coming in and might be at low abundance in our population, this becomes particularly challenging because that means the mutations associated with these variants of concern are also going to be present at low abundance and in a mixture of all the other variants that are also present in the community. And we can't use, I would say, more conventional approaches that you might use for example, a clinical sample where we try to stitch together all the different sequencing reads of the genomes from these samples, because this process, which is called assembly, where we stitch together all the individual reads to try and generate a consensus genome, has an averaging effect. And that averaging effect means that whatever the dominant lineage is in your community is going to basically become what emerges after this assembly process. And so it actually results in a loss of important diversity information, especially when you think about we're trying to detect the emergence of new variants into communities. So we we developed a pipeline that focuses on something that we call quasi-unique mutations. And very simply, we define quasi-unique mutations as mutations that are present in the majority of our variant of concern or lineage of interest, but not present in the majority of all other lineages. And then for each variant of concern, we can establish quasi-unique mutations for that variant based on available clinical sequencing data. And then we can screen the wastewater for the presence of these quasi-unique mutations. So here I'll show a heat map, an example of some of the data where we have applied this approach to detect Omicron in wastewater. So this heat map is showing all the different sampling sites on the y-axis, and then all the quasi-unique mutations on the x-axis. And the different colors indicate whether or not we detected a sequence that supported the presence of that mutation or not, and then the frequency of the frequency of detection of that mutation. So basically, red means we detected every single read that we saw there had that mutation, versus yellow means just a few did, and blue means none of the reads that mapped to that location on the genome contained that mutation. And then white is we had no data, so we couldn't call or not call mutation. So big takeaways here is one, this is actually a pretty good week, but the data is relatively sparse. There's a lot of white here. We don't get complete genomes necessarily, or coverage across the entire genome from wastewater samples. But what we have found is that even the detection of just a handful of quasi-unique mutations has been indicative of the emergence of variants in our communities. And we applied this, we had this pipeline in place before Omicron was introduced in our community, and we actually detected these quasi-unique mutations for Omicron at 6 different wastewater treatment plants before any known clinical cases. So this is just an animation showing over 4 months the emergence of, I don't know, I'm going to do this one more time, the emergence of Omicron across Houston, where we detected it in our sequencing data from the wastewater before any known wastewater cases. And we published a report with other folks across the country that also used wastewater surveillance to detect the emergence of variants in communities prior to known clinical cases, kind of as in collaboration with the CDC, as a demonstration of wastewater as an early warning system. So this information, the sequencing, the levels, it's reported out weekly to many different stakeholders and used to inform action through our health department. It goes to health department leadership, it is briefed to the mayor, it goes to outbreak teams that directly communicate with schools, nursing home, homeless shelters, the jail where we have point surveillance. They've used the wastewater data to actually decide where to prioritize for vaccinations at school. They've done a couple of different vaccination clinics where they've hosted, made available both SARS-CoV-2 vaccination, also influenza vaccines. They did a webinar for school nurses to talk about how to communicate these results and the value of them and whether or not this was providing useful data to them, have them have an opportunity to provide feedback on whether this was a useful source of information on disease spread in their communities. And then we also make it public on a dashboard every single week, so you can look at your wastewater sewer shed, and if you don't know which wastewater treatment plant you contribute to, as most of us probably don't, you can actually go in and search by zip code level. So you can enter your zip code and see, are the viral levels increasing, decreasing, or plateauing week over week. And so everyone can look up, and you can also look at variants. You can see where are we detecting variants across the city as well. And so I'll briefly end by saying that we're thinking about the future of this program, and we're envisioning, this entire system was built up on a single target, right? One single virus. But there's so many other things in wastewater that we could potentially monitor to understand disease transmission in communities and provide an entirely new piece of information on diseases in our city that we didn't previously have. So we've already started influenza monitoring, so all the places that we monitor for SARS-CoV-2 in schools, we also are now assaying for influenza, and we actually are seeing influenza increase across Houston, and we're seeing it in our schools as well. We are starting to look for antimicrobial resistance, and then we're also using some untargeted methods where we're basically just trying to see what all can be detected in the wastewater. not targeting a specific one, just saying what's here, what's in our nursing home samples versus our school samples versus our wastewater treatment plant samples. And we'll use that information to design more targeted approaches if we want to get more quantitative information. And I hope, maybe one major takeaway is that this is, as a tool for public health, is here to stay. I think one of the silver linings of this pandemic is that we've demonstrated this approach at a very, very large scale, not just here in Houston, but across the country and across the globe. And I kind of lifted the text from this slide from a CDC presentation where they talked about what they envision for this at a federal level long term. And so they're trying to support wastewater surveillance all over the country and fund health departments to do this work. And they envision having a core surveillance and emergency surveillance and kind of a special studies arm of this in the future moving forward. And with that, I'll just acknowledge there have been so many people who have been critical to getting this system up and running and doing many different projects that have demonstrated its success. And I'll also acknowledge our funding support and my research group here at Rice. I'm happy to take questions after Dr. Egan's.
All right, thanks a lot, and excited to follow up on Dr. Stadler's interesting work. I'll be using a term called eDNA, which is quite similar to what Dr. Stadler was talking about as well, but we won't be thinking about microbes, we'll be thinking about what I call macrobes, or larger organisms that are not the size of a virus. Likewise, before I get started, I'd like to acknowledge everyone that works in my lab or that I collaborate with. Our group does something called team-based science, so huge network of people. If I use a we in this conversation today, it's a royal we. Graduate students, undergraduates, postdocs, early career researchers, and senior folks that all collaborate with us. Today I'll specifically highlight some collaborations with some nanoparticle physicists at Notre Dame that I collaborate with and have for many years. I'll also focus on a former master's student, Jen Drummond, who Dr. Adrian Correa in my department and I both co-advised. And then I probably won't have time to talk about some work by Pedro Brandao in my lab in collaboration with Jen Tank at Notre Dame. So we'll save his exciting work for another installment. And the undergrads and all the funding that contributed to the eDNA work in our lab, specifically the USDA, CERDIP, and a collaborative project I have with Dr. Stadler with Dow, locally here. All right, so eDNA, what is it? eDNA, for me, as a biologist that studies animals, is analogous to forensic genetics. So as we all know, if you have a pet at home, or maybe even children, organisms leave behind microscopic bits of tissue, including slough tissues, scales, hair cells, microscopic larvae, milt, eggs, or even if you're an insect, you might leave an exoskeleton around. So they leave particles in the environment constantly. So these microscopic bits of tissue also contain DNA. The sequence of bases in some DNA sections is unique to that species, and we sometimes refer to it as a barcode that can identify that species. And so I'll talk a lot today about sampling aquatic environments, both freshwater and marine. So in the water column, this DNA is present, but it slowly degrades through time on the order of hours or days. So that also means if we get a detection of something, we have some time scale. We know it was present the last few days. And so how do you sample eDNA for me? So I'm not sampling wastewater, I'm sampling just an aquatic environment. We collect it in some way, we then filter that sample, and then we do a DNA extraction on that filtrate. And that filtrate contains all of those tiny particles that have been shed by all the organisms that are present in that environment. And then we follow up in doing something with its DNA. And I'll talk about a couple different methods today. So for my talk, I'll focus on three components. One, the detection of invasive species that can cause economic and ecological damage. I'll then talk about how to take those lab-based techniques and get out into the field, so field-based DNA detection tools in collaboration with those nanoparticle physicists from Notre Dame. And then lastly, we'll expand out from thinking about detection of single species to surveilling entire biological communities using similar techniques. All right, so first topic, detection of invasive species. What's the problem? So aquatic invasive species cause huge ecological and economic damages, and we need tools to detect them before they get here or when they're still rare in the environment. Why should we care? Well, using the US Great Lakes as an example, there are over 180 documented invasive aquatic species there. They cause something on the order of $130 million in annual damages. They foul boats, they get in the intake and outtake of nuclear power plants, they cause tons of damage. And of course, with an increase in global commerce and movement and very little ability to detect these things, this problem should only increase. So we wanted to explore whether this environmental DNA technique can serve as an effective detection tool, especially for things that are really, really hard to see using traditional methods. And I'll focus a lot of this example using ballast water from large ships. So if you're not familiar with this, so transoceanic ships, these giant things that come in and out of the Houston Ship Channel every single day, they have these things called ballast tanks. And so as they unload or load their materials, they have to move water in and out of these ballast tanks so they don't topple over and fall over. So they take water off the coast of one continent, and they come, and as they unload, they dump that ballast water off the coast of another continent. A huge opportunity to move invasive species across the planet. And I'll focus this first study on these 5 different critters who are known to be passed through ballast tanks of these transoceanic ships. 2 of them, zebra mussel and quagga mussel, are invasive species already here in the United States, and the other ones are suspected to come here because the US environment is similar to the native environment where these organisms live. All right, so again, this method, we'll kind of adopt this method to think about how this can work with these invasive species. We collect water. For the first thing I'm going to show you is just a simple experiment in the lab where we'll basically take a very, very small piece of the tissue of these different organisms, place it in that water sample as if it was collected in nature. We'll filter the water, extract the DNA, and then we'll do some amplification of that DNA through PCR and just simply look to see if an individual is present and detectable in these samples. So here you can see just a gel electrophoresis technique where we just simply amplified a specific gene in each one of these species. We ran it out on a gel. If that tissue was present, if we detected the DNA, we should see a big bright band in those wells, and in fact we do. So traditional PCR in a lab that we teach our undergrads can detect species of concern in a aquatic sample. Okay, so that makes sense. So then we wanted to kind of go out and expand from lab and also from these organisms. How broadly applicable is this? So Daphnia are an invasive species in certain regions of the US. Rusty crayfish is pushing out native crayfish in the Midwest. And Corbicula is an invasive freshwater clam that's displacing native clams in the Mountain West. And in addition to expanding outside of taxa to see if this worked, we also wanted to move from PCR, which gives us this kind of detection, yes or no, to something a little more quantitative. So we started off with qPCR, quantitative PCR, which uses some fluorescence so that you can actually back calculate the original starting concentration of the DNA in a sample to allow you to predict the abundance of that organism, the biomass of that organism in nature. And so we can see things. So now looking at these 3 panels I've shown here, just the x-axis is time, and the y-axis is the kind of DNA copy number calculated with qPCR. And we did, again, some experiments where we added Daphnia or rusty crayfish or Corbicula into some aquaria and just took samples through time. And you can see already at day 2, we immediately can detect that organism. without collecting the organism and just detecting it in the water it swims in. And then when we totally remove all organisms from the aquaria, we can still detect that DNA present in the environment for a couple days past that. So it accumulates and then it quickly degrades. Okay, so it works in lots of different organisms. It works as both a detection tool and potentially quantitative, because we can actually see it increase through time. We also can see it increase with biomass. So we've now gone out into nature to explore this. So in the top left, we've looked for Daphnia samples, not by sampling the Daphnia themselves, but just by taking a cup of water and filtering it. And we find that we can detect them on the bottom of certain ponds, offshore or even on the shoreline, but that the detection differs by the organism we're trying to study. In the center panel, we've also expanded to look at this invasive rusty crayfish. We've looked at a series of lakes that have varying degrees of abundance within them, some high, some low, and we can again see as crayfish abundance increases, our detection using qPCR also increases the number of copies of DNA we find in solution. So across lakes or even within lakes works. And then last on the bottom, we have this invasive clam species, Corbicula. There's a huge patch of it in this one river. We sampled around the river, and then we went downstream a couple kilometers, and we can see how the copy number, our ability to detect it, decreases through as it flows away from that kind of source patch. So it works in the field. It should work for some of these detections of invasive species, which is really nice. So I'll go back to my ballast tank example that I started with, because of course a lot of this is involved taking a sample and going back to a laboratory and using expensive equipment that requires training to be able to really get these detections. But is that really going to work for someone who works for Border Patrol or the Coast Guard, and they're actually trying to intercept ships that might be bringing invasive species? And from the business side of things, no ship is going to want to just sit off the coast of Texas for 3 days while we screen their ballast. Okay, so could we develop something that's more real time? So that brings in my collaborators, Carol Tanner and Steve Ruggiero, who are physicists at the University of Notre Dame. And they have the ability, they've designed this tool called LTS, or laser or light transmission spectroscopy, which basically allows them to measure the size and the density of really, really small things on the order of nanometers. And while they were measuring gold particles or some other purpose, our group thought that we could combine their technology with some environmental genetic sampling to be able to build a detection tool that could work in real time in the field. Their device is small, it can run off of a car battery or even a solar battery, so you could effectively bring it in the back of a pickup truck or even onto a ship. All right, so the way this works at the nanoscale is they're measuring the size of some small nanobeads, which I'm showing in the top left, and our idea was to functionalize those nanobeads with a single strand of DNA that would complement a target, so it would only bind to a target or dangerous species, it would not bind to others, and thus it would grow that tiny nanoparticle by some degree, versus when it was in the presence of other DNA and it would not grow. So growing because it binds to the target versus the non-target. And if we could detect those differences, then maybe we could design a tool that would be more user-friendly in the field. Now, the way their device works is they shoot a tunable laser through two solutions, one with our sample of nanoparticles that has been hopefully bound to its target if it's present, and then a reference sample. They then compare those and look at how light by wavelength goes extinct through the solution, and they effectively can calculate, using the extinction curve, the size and the density. So if the target, the dangerous species, is not present, we should see a single distribution of the particles, and if there are some target present, we should see a shift in the size of those nanoparticles. That would be a signal that the dangerous species is present in that solution. So we applied this back to our ballast tank experiment, thinking about these invasive species. And now we're going to use a universal set of primers that will amplify the DNA of all of these organisms in a single reaction. And it's the nanoparticles themselves that have the diagnostic fragment that will either bind or not bind to each species. And here's what we find. So on the top center, you can see the nanoparticles before anything in one of our controls has been bound. And then in the presence of the golden mussel, you see there's a shift. So some of those nanoparticles do bind to the golden mussel, and they do show that size shift. And in fact, I have an exemplar of each one of the 5 invasive species, and in each one you see some size shift of these nanoparticles when they bind to the target species. So this worked. We were really excited about this. And we repeated this over and over and over, and we found that there was just on average this nice shift of this second peak when you see these present. So this should work in the field in real time, which is quite exciting. But of course, we're still in controlled environments, so we wanted to expand and get a little bit outside. So we actually did run it off a car battery, and we went out in the field to sample this. We went to a lake in Columbia, Missouri, which was confirmed to not have zebra mussel at the time. And then we went to a lake in Michigan that we knew had zebra mussels, the invasive species zebra mussel present. And we found that detection in the one where we predicted it, and nothing in the lake where we did not think it was present. So cool, so it seems to be working in the field, works in these ballast tanks. Well, let's actually go onto a giant boat. So we actually collaborated with a shipping company. They let us board this 1,000-foot shipping vessel. We boarded in the middle of the night in northern Indiana, in the town of Gary, Indiana. And we took samples. So they bring in the ballast or release the ballast as they're taking on or releasing. I think what they were doing here was some sort of ore, like an iron ore that they were putting in on the ship. And then we followed it as it traveled through Lake Michigan, up into Lake Superior, into Duluth, Minnesota, and took samples along the way. And just like with our examples from the past, now focus just on one invasive species, quagga mussel. So first, here is a negative or a control where we see a distribution of these nanoparticles functionalized with the single-stranded oligonucleotides that would bind to the target. We see one distribution. And then now with the source harbor where they're taking on water for the ballast tank intake, so they've taken this water into the ship, from the ballast discharge where they release the water back into another harbor, and then the receiving harbor, we were able to detect quagga mussel in each and every one of those sources. So pretty clear evidence that this technique, this tool, could be really effective in actual real time to detect these invasive species. So we knew quagga mussel was present in this environment. We knew it was in both source and receiving harbor, so none of our data was a surprise, but it was a nice proof of concept for how this would work. All right, so the last part of my talk will expand out from thinking about individual taxa and detecting individual dangerous species to now thinking about whether we can use environmental DNA to sample or surveil entire biological communities. And this has been published in a paper by a former master's student, Jen Drummond, co-advised by Dr. Correa and myself, and a bunch of collaborators. So the goal of this project was to kind of elucidate spatial patterns in the distribution of plant communities associated with a set of lakes in the upper peninsula of Michigan. And we're going to use something called eDNA metabarcoding. So before we were amplifying a very specific gene and first asking if it was present, then using qPCR to ask what the copy number that was present in our samples. Now we're going to go back to those universal primers that will amplify a certain gene region for an entire group of organisms. This one's going to amplify a gene in plants. And we're going to ask the question, now throwing that sample onto a Illumina sequencer, which can sequence many different fragments in parallel. We call it high throughput sequencing, so now just not a single sequence, which we call Sanger sequencing, but generating on the order of 10 million reads from an environmental sample from one of our eDNA sampling points. All right, so we're sampling these lakes and asking questions about whether eDNA can characterize the plant communities in and around these lakes. All right, so I just have a nice little kind of diagram of how this works. So again, the DNA metabarcoding, what we're doing now, we take the sample, we extract DNA from that filter, and now we're going to amplify all of the different organisms present for the exact same gene so that we can then identify individual species if that gene region is diagnostic for that species. So we sampled locations across all 6 lakes, both near shore and offshore, both at the surface and at the bottom, very deep. And so we have this nice little matrix to see where our eDNA samples were taken, to see if they differ in the communities they detect. And so here's some of the actual results. So we now have all of these tens of millions of reads. We can compare them to databases of known sequencing reads from these same genes, and thus identify the fragments we have in our sample. And for those that we were able to identify, we were able to categorize them into 3 broad groups. Aquatic samples, we predicted they were there. Wetland samples, so these are things that are growing out of the water but near and associated with water. And then true terrestrial things that are growing more in uplands, so things like a pine tree that you can detect from the water sample. I'll focus your attention first on the center graph. This is just simply the number of read counts we got at different parts within the pond, so offshore versus near shore, at the bottom versus the surface. And you can see that the vast majority of the things we get are aquatic organisms, so that makes sense. However, if you just ask the question, which of those reads identifies to a specific species, you can see that over on the left, the actual taxon counts. And what's really interesting is that, of course, in the blue, we do sample many aquatic species. In the green, we sample many wetland species. But I was really excited to see that we do sample a significant degree of the terrestrial community that surrounds these environments. So sampling the water is a nice way to sample the entire environment in a given region. Okay, so now another exciting result broken out by the 6 lakes was just a measure of species diversity within each one of these lakes. And what you can see is, while there is some variation here, on average, each sample tended to have a similar degree of diversity sampled, and that was, in fact, statistically clear. The only difference was there was a very subtle difference between our nearshore samples and those that were offshore, but that difference was a very, while significant, a very subtle difference. However, even though diversity was similar, the different communities were dissimilar. So in the plot on the right, we've now mapped using something called an NMDS plot, which basically each dot is a sample, and the differences or distance in these plots between dots is basically the difference in the number of different types of species present. So if they're very close, they have a similar community, similar set of species present. If they're far away, they are quite dissimilar. And they found, we were able to find that lakes differed kind of ecologically. Some were shallow, some were deep. Those that were similar overlapped with each other, and those that were ecologically different, had different sets of plants present in that lake, were in fact different. So that was really exciting. So while diversity was similar among samples, the types of diversity sampled was different based on the ecology of each one of these ponds. That was really exciting. And then if we look at these same type of NMDS plots for any of the other metrics, near shore, offshore, bottom versus the surface, we found that they're very similar. So the big conclusion here for conservation biologists that might want to sample these ponds and not be an expert in every single aquatic plant present is that the easily accessible shore-based sampling tends to be a reliable technique that can inform conservation, research, or management of those different ecosystems. All right, so I hope in my follow-up to Dr. Stadler's work, I can show you how we expanded from thinking about tiny organisms to being able to sample tiny pieces of much larger organisms in environments, to be able to sample things like fish species, to detect invasive species. You can also think of other rare projects such as an endangered species that's quite rare in the environment, or just things that are really hard to find in the environment using traditional methods. eDNA tends to be a really nice tool. And with that, I'll again acknowledge everybody and the funding with my group, and then I'll invite Dr. Stadler back up here so we can answer questions.
Wonderful. Thank you for two fascinating, very timely talks. Opening the floor for questions. Question?
Hi, this is for Dr. Stadler. I was curious about, you sample the wastewater treatment plants, and you also sample individual sites like hospitals or schools or whatever. Are those data combined into one set, or are they used for different reasons, different purposes?
So they are used right now for different purposes. So the wastewater treatment plants, because they cover just so many more people, those are used to kind of get a sense of what's going on at the citywide level. But the individual point surveillance, we actually analyze with a slightly different like statistical model where we basically threshold it. So we have different thresholds for whether it's positive, inconclusive, or really it's like inconclusive, positive, or high positive. And then we basically just, for each of those point surveillance spots, like a nursing home or a school, those individual results each week are communicated back to the facility, and they have different action plans depending on the facility. But they're usually for those point surveillance ones looking at whether or not it's positive or high positive, and especially if it's for multiple weeks at a time. And so a lot of it's like, it used to be, can we get testing to these locations? So can we, it was mostly to like inform, okay, here's your local testing center, here's free testing, let's put a mobile testing in here to see people test it. Now it's okay, here you can be here's where you can get vaccinated. If you're not vaccinated, they have metrics at each of the schools where they're actually tracking like percent vaccinated in that community, percent vaccinated. So they have targets they're trying to reach in each of these schools and each of these specific communities for vaccination rates. The schools were selected originally because they were located in high positivity rate areas of the city. So they tend to have lower vaccination rates actually. So they really use it for like targeting resources to get vaccines or get information to these communities.
Another question?
Keep your microphone. For the wastewater testing, I'm assuming these are tests coming off collection lines before it has reached the wastewater plant?
Oh yes,
And not after it's gone through the chlorination
correct.
beyond the
Correct.
Clarifier.
It's untreated wastewater. So the treatment process, the wastewater treatment process, results in like almost complete elimination of the signal. There have been a number of people who have looked at this. So some of the viral particles can pass through the wastewater treatment plant, but if you look at influent versus effluent, there's a drastic decrease. So if we're trying to detect kind of what's in the community, we're grabbing untreated wastewater samples. So before it's gone through any wastewater treatment.
What is the lag time between grabbing the sample and having the results?
So right now so this obviously is a very large operation at this point. So samples from the wastewater, we get different sets of samples at different days. For the 39 wastewater treatment plants, the samples are collected from Monday at 7 a.m. to Tuesday at 7 a.m. So they're 24-hour composites. And then they are collected by the Houston Water folks and delivered to our lab on Tuesday morning and the health department lab. We have results reported out by Thursday. We probably could report them out faster if we really wanted to work like around the clock, but that's how we've operationalized it. And then we have sequencing information usually by Friday afternoons. So we'll have the quantity of the virus in the wastewater by Thursday, and then the sequencing variant screening reports usually by Friday, sometimes Saturday.
That is still pretty fast compared to what all the other things that are being tested in the wastewater. Some
Right.
of those don't even get.
And I think kind of the future for this will be like even faster and even easier. easier. So I think, one future application this will be like similar to like a home test where you could, not for a home, but for a building maybe, where you like are having a really easy to use assay that you could do like presence absence with. It might be more crude and slightly less sensitive and not quantitative, but you could, let's say a school, our vision for this would be like, okay, a school janitor could like grab the wastewater in the morning, assay it, and just see if it's positive or negative, or any other number of viruses, right? I mean, as someone with like young children, I would love to know if like RSV is circulating in my kid's daycare. That might impact my decision to send them to school. I don't want to hear about it after 3 people have been tested, from a doctor after it's taken 3 days. It's been circulating in the community. So I think the vision for this is if this were easy to use, easy to apply, this information could be like a lot, very effective at curbing spread of lots of diseases.
Also for Lauren Stadler. I was wondering if this kind of research is done all over the world, or is it like a first world thing to do only, or can you use this in, India or Africa or someplace where you don't have a huge amount of public resources and still get really good results and maybe better results than other kinds of ways of finding out where the virus is?
So that's a great question. I think, so first of all, it is definitely being done all over the world, and it's kind of amazing how many groups across the world have been working collectively across the pandemic to establish these methods and this approach. I actually think one of the real advantages and the power of wastewater monitoring is it can actually be used in underserved communities or places that are low resource to prioritize, right, where to spend resources. That being said, most of the applications that have developed it and implemented it at large scale, they started probably in more like, where you have advanced labs where people could develop these methods, or you have a lot of facilities available. But now it's everywhere, it really is. It's, I think, on every continent. There's this website called COVID Poops that aggregates all the different sites across the world that are currently using wastewater monitoring. There's like thousands and thousands of sites, and it's really incredible. And I think the vision, again, for this is this becomes a surveillance tool, a global surveillance tool, where let's say someone in India detects some new virus or variant in a wastewater that could be quickly disseminated to this network of people working in this space, and we could all be looking for it, right? And that's actually how COVID, a lot of COVID variants, that's how this has worked. But when we used it and applied it to environmental surveillance, we could have an even earlier warning system.
I have a question for Professor Egan. So it looks like in your eDNA work, you're sort of testing for known organisms where maybe they've already been sequenced or at least partially sequenced. I'm sure you know and can maybe comment on the Venters or C eDNA experiments where they discovered there were, a gazillion species, right? So my question for you is, can you be on the lookout for new species in your work?
So yes, thanks for the question. In the eDNA metabarcoding approach, where we're just sequencing anything in a community for a given gene, that's where we come across, I like to make the analogy between dark matter and dark organic matter. So we actually get many more sequences that are currently not matched up than are matched up. And that's for 2 reasons. One, we don't know everything in an environment. We know that even from the Rice University campus, where my lab has discovered new species on the oak trees out the front door of this building. But we also don't have the genetic resources for every single species on planet Earth right now. So we have a little bit of a mix and match of whether we can find that. So yes, we see things that we don't know what they are yet. And it's really exciting when they are extremely different than everything else we know, because that suggests that maybe a new lineage. But it requires some follow-up, because at that point we just have a fragment of DNA. Question.
So I also have a question. You keep the microphone. Okay. So really impressive operation, just to think about the coordination with your collaborators. And so my question is about scalability. And so you're relying on ballast because that's how these species might be transported, and that's also where you're sampling from. And you're in a lake system, big lakes, great ones. So I'm just wondering if you can comment both on the scalability of your sampling process and also on the scale of bodies of water that you can test.
Right. Yeah. So we started off even less than lakes. We started off in buckets to explore how this would even work, and we were trying to get bigger and bigger. So we have a current project funded through CERDIT, through DoD, to explore river systems. And of course, if you're at the mouth of a river that's flowing to the ocean, you are sampling potentially the entire watershed. And so if you know anything about how DNA degrades through time, the speed and velocity of that water, then you can back calculate up that watershed and slowly move up to sample something. So that could work. Here in Texas, we've thought about applying that to endangered species, where there's small amounts of public land that we can sample on, but that would, if we find the target organism we're looking for, it would suggest it's up this area, which is private land that we can't sample, but we at least know they're present and happy on those private lands. We are expanding into the ocean, so I have a collaborative project led by Dr. Adrian Correa, where we're looking for the effect of invasive seagrasses that have brought in with the increase of hurricanes and how that's impacted the local fisheries. So now we're sampling the presence of one species and then seeing how that impacts an entire community of fishes that tend to, their juveniles live in the grass beds, and maybe invasive species and native species are different types of habitats. So we're working at the scale of coastlines at this point, and rivers, but we're not looking quite yet. The other project I've worked on that's a little bit broader in scale is we've sampled ports across the world and looked at the local native diversity versus the presence of very common invasive species, like some of the ones I was focusing on today. And what we're seeing is that the more traffic that a port has, the more homogeneous those communities are. They're just more similar because invasive species are being moved into them. So, you're still taking samples at a point of contact, but you're just limited by the money and the number of samples and people you have to do it.
So it says, if we look at a general environmental sample, freshwater or wastewater, and sequence it without targeting a specific species, is the genomic landscape dominated by macroscopic or microscopic species genetic material?
Microscopic. And we could even start with the human body. There are more microbe cells in our bodies than human cells, right? So that just scales. So microbes, I won't say this much, but they're pretty important.
Wonderful. Well, thank you both so much, and thank you for the great questions. And those of us in person, please join us in the lobby for the reception. Thank you.